membrane. So I've posted this PowerPoint. I have on here that the important information is in black. I have given you additional information in red. The additional information is, you don't have to pay me any extra for that. That's just for you guys. You will be expected to know the information in black though. Kate, you're just supposed to be following along right now, but you don't have to fill out any handout. If you've printed it, feel free to highlight things as we go along. Okay, so I want you guys just to listen. Okay, awesome. All right, so we talked about yesterday the fact that you guys all started off as one single cell. You guys learned about this in your style lesson as well. So the egg and the sperm come together. That creates a brand new single cell that has never been seen on this planet before. Your DNA combination is unique to you. You got half from your mom, half from your dad, but every single time sperm and egg come together, it's a different combination of half of the DNA from the mom and half from the dad, making a unique individual. We can talk about twins later on. That's maybe a side note here. Now you guys know you're made up of over 37 trillion cells. So we're going to be looking at how does a single cell organism develop into a fully grown, complex multicellular organism. There are 3 reasons why cell division happens. One is for reproduction. We're going to talk about this in a second briefly. Mainly, we're going to be talking about cell division in humans for growth and to repair damage. So you guys are probably approaching the point in your lives where you will actually slow down and stop growing, but cell division is necessary for growth. And it is also essential to repair damage and to replace dead and dying cells because we know that happens all the time. But let me focus on reproduction for a second. Not all living things reproduce the same way. Humans reproduce slightly different than some of the other members of the animal kingdom. There's some similarities, but we reproduce obviously very different from the plant kingdom. So reproduction is essential for life to continue, but not all organisms reproduce the same way. There are 2 main types of reproduction. One is called asexual reproduction. The prefix a actually means without. So without sexual reproduction is when you have one parent that has offspring that are genetically identical to the parent. So asexual reproduction is similar to the concept of cloning, where you have one parent and all of the offspring are genetically the same as that one parent. Does anybody know what organisms or what would be an example of something that goes through asexual reproduction? Yeah, Kate, some plants will go through asexual reproduction. Pretty significant in our lives right now is bacteria or viruses. You have one parent, and over time that grows and splits into 2, and then 4, and then 8, and then doubles every time that there is a division cycle. Obviously, that's not the way that humans or most of the animal kingdom reproduces. Sexual reproduction, and this is where we're focusing mainly in our discussion today, is 2 parents having offspring that are genetically different to either parent. Genetic tendencies are similar to those parents, but the offspring are genetically different to the parent. Any questions on either what asexual reproduction are or sexual reproduction are, or any questions you guys hear about this have about this topic? So one thing I want to point out here before we move on, if you guys do continue with biology, you'll learn about a process that's called meiosis, and that's about how gametes or sex cells are formed, so egg and sperm, in order for sexual reproduction to happen. But your skin cells growing and making copies of themselves is actually asexual reproduction. You have one skin cell and it can grow and split into 2 skin cells, or your muscle cells reproducing and growing, or cardiac tissue. So cell division is actually a form of asexual reproduction. One parent having offspring that are genetically identical to that one parent. Okay. So as organisms grow, as you went from a baby to a toddler, so on and so forth, up to a teenager, you grew because the number of cells in your body increased. Your cells did not just get bigger, the number of cells increased. And that process, it happens at different rates depending on the types of cells in your body. We already discussed that every day, millions of skin cells on your body die and need to be replaced. And also other types of cells in your body die and need to be replaced. You guys already know, according to the cell theory, that all cells have to come from pre-existing cells. They don't spontaneously come from nothing. So we're going to be focusing on that process that allows the body cells to reproduce so that we can maintain a healthy overall organism. This is a quick picture of the cell cycle. So if you start up here at the top, you've got a cell, the cell cycle starts, the cell grows. We just talked about this. You're going to have your mitosis, your organelles replicating, and then it moves into this process of mitosis, which is just a portion of the cell cycle that is focusing on the nucleus dividing, because that's critical with your DNA inside. Once the cells get ready to divide, then it splits into two, and those two cells immediately go back into the cell cycle. It grows, it receives instruction from the overall organism, the DNA replicates, the cell gets ready to divide, so on and so forth. And this keeps going constantly in your body at different speeds, obviously, depending on what type of cell we're talking about. But on average, every 24 hours, one cell splits into two in your body. We're going to be focusing on just this small segment. It's actually about 10% of the overall cell cycle is the process of mitosis, which is the nucleus dividing. Any questions so far? There are three main stages to the cell cycle. The first one is called interphase. 90% of the cell cycle is in interphase, mitosis, and then cytokinesis. Does anybody know what the prefix cyto means? You guys saw it in like cytosol as part of like your cell organism. Sophia, do you know?
I have a question.
Okay, hold on 1 second. So cyto means cell, and what does kinesis mean? If something is kinetic, what does that mean? It means movement. So right here, cytokinesis means the cells move or they divide and split into two. So the three main portions of the cell cycle are interphase, mitosis, and then cytokinesis, and then it starts all over again. Sophia, what was your question?
You just answered it.
Oh, okay. Now, let's focus on what's happening right here in mitosis. There are 4 phases in mitosis. It's called prophase, metaphase, anaphase, telophase. If you remember write this on your notes if you printed the PowerPoint. If you remember the acronym IPMAT, I-P-M-A-T, IPMAT, it will help you remember the order of the phases of mitosis. Interphase is the beginning of the cell cycle, interphase, and then prophase, metaphase, anaphase, telophase. So just jot down IPMAT somewhere in your notes. I'll put that in your chat so you know the order. IPMAT.
How do you spell like metaphase and stuff?
Oh, I'm actually going to show you, Lila. It's coming up.
And what's IPMAT for again?
It's an acronym to remember the phases in order. I'm going to show you guys here. So if you hold on a second, I think it'll become more clear. All right. So here we go into lots of information about what's happening with cell division. Okay, so I stands for interphase. It's actually not a part of mitosis. It's pre-mitosis. These are the things you guys were mentioning that have to happen before the cell can just randomly split into two. So there's some key things that happen in this phase. That's the longest phase. The first thing is the cell has to grow in order for there to be enough cell material to split into two fully functioning cells. The second critical thing that has to happen in interphase is that the DNA has to replicate. Why is that important, that the DNA makes a copy of itself?
So there can be like a new nucleus?
Yeah, you have to have a fully functioning nucleus, the control center, in your new cell, so the cell knows what it's supposed to be doing. So the DNA makes a copy of itself. Initially in interphase, the DNA is in really long invisible strands called chromatin. So the reason why it's long and thin is so that it's completely stretched out and every portion of the DNA can get replicated. If it was all clumped together, maybe not all the DNA would get copied properly. So in interphase, the DNA is in long, thin, invisible strands called chromatin. The way that I remember this is, do you guys, can you picture this, or have you done this as a kid, where you have a tin can with a string going to another tin can and you talked to your friend or your sibling on the tin can, the sound vibrated down the string? Has anybody ever done that? Maybe you need to put it on your to-do list for the weekend. Chromatin is long, thin strands. So if you can picture the tin can with like the string to another tin can so you can talk, hopefully that will help you remember. Chromatin is long, thin, invisible strands of DNA, and it gets replicated. Some of you guys are saying in the chat, you have seen this in pictures of like the olden days in black and white, like me when I was a kid. Okay, the next thing that happens in interphase is that the organelles have to be replicated so that there's fully functioning mitochondria and Golgi bodies and vacuoles in the new cell. So basically, if you look at this list, everything is doubling or replicating here in interphase. The cell gets bigger. The DNA makes a copy of itself. More organelles are made. And the last thing that happens is that the cell has to wait for the body to say, yes, please go through cell division. What would happen if there was no speed regulator on our cells? What would happen if our cells just kept dividing and dividing and dividing and dividing?
Can't that lead to cancer?
It could lead to cancer. Yeah, it could lead to cells that are just not benefiting the overall body. Or in essence, we just have a lot of cells that aren't necessary, that all they're doing is just using up energy and nutrients, right? So you could see that there's a reason why there needs to be a bit of a speed regulator. If your body gets cut, it's like, go, go, go. We got to fill in this cut, you know, repair this damage. Or if you have an illness and you need some more white blood cells or something to fight your sickness, your body says, go make those things. Otherwise, it could be really chaotic. So there's a bit of a speed regulator that's saying, okay, now it's time to continue going into the process of mitosis. Any questions on interphase? So again, this takes about 90% of the cell cycle. The cell is growing and replicating so that it's ready for when the body needs it to split into another copy of itself. Next is prophase, and this is a picture of just the nucleus here, this computer-generated image. Over here is actually a slide of an animal cell focusing in here on the nucleus. You can see that this is representing my DNA, or all of the strands of the blueprint here in the middle of my nucleus. There are a lot of things that happen in prophase. In prophase, the DNA condenses, so it gets compact and smaller. And the word chromatin changes now into chromosomes. You guys have probably heard of chromosomes before. The chromosomes are your DNA or your genes that make you who you are. Each chromosome consists of 2 identical strands of DNA. So remember, the DNA interface has replicated, it's copied. And the 2 identical strands are held together, and we call each of those strands held together sister chromatids. Can you guys see why there's so many crazy terms in biology? We're going to be reviewing a lot here. Chromatin condenses into chromosomes. Each of the replicated copies are called sister chromatids. Those chromatids are held together in the middle by a sticky middle called a centromere. So imagine like twin sisters all lined up in the hallway like they're going down to a washroom break or something, holding hands. Identical copies of DNA held together, holding hands. Why do you think the identical copies have to be stuck together? Why is that important?
Otherwise, you won't know if they're identical or not unless you like try to find it.
Yeah, exactly. So imagine you've got like a 50-page blueprint of a building you're going to make, and you copy each of those pages, and you need to make sure that each cell or each person gets page number 1 and page number 2 and page number 3 and page number 4, right? the identical copies are stuck together so that we know there's replicated strands of every single portion of that DNA code. Okay, so the DNA condenses. It condenses so that it's compact and organized, and each of the identical copies are held together, and we call those sister chromatids. The next thing that happens in prophase, oh, and I've mentioned this, the sister chromatids are stuck together by a sticky middle called the centromere. The next thing that has to happen is the lockbox around the DNA and the nuclear membrane around the nucleus disappear. Why do you think that happens?
I'm confused on what number 3 means for that.
Okay, hold on one second, Sophia. So if we picture, this is my DNA, this is the stone that I have here, this is my DNA. If I imagine my DNA is stuck inside of this stone, why do you think this outside surface has to disappear before the cell can split?
Is it because like if it's like there, it can't go into like 2, so it just stays as 1, so it wouldn't be in the other one?
Exactly, Kira. Can I get to the DNA right now inside of my nucleus?
No.
No, so I can't separate my copied instructions, my DNA that's in here. So the nucleolus and the nuclear membrane have to disappear. Otherwise, my DNA is just trapped in the middle, and that's no good. It's totally useless if it's held trapped in the middle. So the lockbox and the nuclear membrane have to disappear so that that DNA then can separate and go into the new cells when the cell cycle is complete. So Sophia, nucleolus is an extra layer of protection around my DNA, around my chromosomes. So the nucleolus is like a lockbox, keeping my DNA nice and secure. And the nuclear membrane is the layer outside of my nucleus. So both of those things actually dissolve and just completely disappear. Okay, so if you want to put notes on the PowerPoints that you've printed, the nucleolus is an extra layer of protection around my DNA. And the nuclear membrane, you guys know, is outside of my nucleus, allows things in and out. It's like the gatekeeper to my nucleus. Okay. The last thing that happens is organelles that only form in mitosis, they actually grow, they show up because they have a specific role that they have to do. There's something called spindle fibers that form and centrioles. I'm going to go back to the picture here in a second. Spindle fibers form and stretch from my centrioles. Just hold on to that for a second. I'm going to go back to my pictures of prophase. Do you guys see these things that look like marshmallows on the end here? These are my centrioles. Think about them as like poles, like north and south pole. Centrioles form, and these spindle fibers are growing out of the centrioles. I'm going to tell you what they're going to do here in a second. But in prophase, the DNA condenses, my sister chromatids are held together, identical copies of my DNA are all organized nice and neat. The nucleolus and the nuclear membrane are going to disappear. My centrioles form, and these spindle fibers start to grow. All of those things happen in prophase. Questions about prophase?
So sorry, what's the difference between the nucleolus and the nuclear membrane?
The nucleolus is an extra lockbox around my DNA. So the nuclear membrane's outside of the nucleus, Lila, but the nucleolus is around just the DNA. It's an extra layer of protection, keeping my DNA secure and safe. Okay. All right. So I want you guys to look at this next picture. This is metaphase. What do you notice that's different about this picture compared to the one we just saw?
The spindle fibers are like longer and attached now.
Awesome. Kira's noticing the spindle fibers have grown, and they're actually attached to each of these centromeres in the middle of my chromosomes. What else do you notice?
The DNA are like the red and the blue.
Yeah. Where are they, Sophia?
in the middle, whereas the other one was kind of like spread out everywhere.
Perfect. So if you guys even look at my cell picture here from the microscope, all of my DNA is lined up right in the middle of the cell. So if you want to highlight the letter M in metaphase, my DNA lines up in the middle of the nucleus in metaphase. That's literally the only thing that happens in metaphase. The spindle fibers grow and they attach to the centromere. So imagine the DNA is like a whole bunch of twin sisters all lined up holding hands. These spindle fibers are growing and attaching to their hands that are being held together, and they're lined up orderly right down the middle of the hallway. Okay, so spindle fibers attach to the centromere, and they actually organize the copies of DNA right across the middle of the nucleus. What do you think is going to happen next? What's going to happen to my copies of DNA lined up in the middle of the hallway? What's happening in this picture?
They're separating.
Yes. So in this picture, you can see that the twin sisters, the sister chromatids, have been separated. One copy is going one way to one pole, one copy is going the other way. What is actually pulling them apart?
The spindle fibers.
Yes. So the centrioles, the poles, have these spindle fibers that have grown out and attached to the centromeres. Now the spindle fibers are shortening. There's actually motor proteins on the spindle fibers, and they're literally like pulling them in. Okay, the motor is like rolling up those spindle fibers, pulling those chromosomes toward the ends, because eventually you're going to have one copy of DNA at one pole and one copy of the DNA at the other pole. So remember, if we think about this like a whole bunch of pages of my blueprint, page 1's going left and the copy of page 1 is going right. Page 2 is going left and the copy of page 2 is going right. It's separating out the copies of the DNA. Okay, so here's what I have listed for anaphase. The spindle fibers are contracting and shortening. The motor protein is pulling the rope closer to the centrioles. That separates the centromeres. And now that the sister chromatids have been yanked apart, we conveniently changed the term one more time, and now they're called daughter chromosomes. So we only call them sister chromatids when they're actually holding hands lined up in prophase and metaphase. Now in anaphase, they're being separated and we refer to them as daughter chromosomes. One set of daughters goes one way and one set of daughters goes the other way. So the chromosomes are being separated. In essence, the daughter chromosomes are being pulled to opposite poles to each of the centrioles. Oops. Anna, this probably doesn't help you, but Anna actually means back. The prefix Anna means back, so they're being pulled back. But I like to think about it as A, away. So first, the copies of DNA were in the middle in metaphase, and now in anaphase, they're being pulled away from each other. So if you guys want to write that in your PowerPoint notes, highlight the A and say away. The daughter chromosomes are being pulled away and separated, and they're going to each of the opposite ends of the nucleus. Questions on anaphase? I know it's a lot, guys. We'll be reviewing this as much as possible. What do you see happening here in telophase? Or sometimes it's called telophase, depending on where you are from.
It looks like the cells are being, like the cells splitting apart with the separate DNA.
Awesome. You can see there's one set of DNA here on the left, one set on the right. What do you see forming around the DNA?
Is that like a nucleus?
Yeah, so the nuclear membrane's regrowing here. The nucleolus will form, keeping that DNA nice and secure as when the cell splits. And Lila was saying it looks like the cell is starting, the nucleus here is starting to get ready to split. And what's happened to your centrioles and your spindle fibers?
Are they like shortening?
Yes, exactly. They've shortened and pulled that DNA package all the way to the one side so the cell can split in a nice organized fashion with one set of the blueprint here and one set of the blueprint here. If you guys want to write on your note that telophase and prophase are opposites, it actually helps you to memorize this faster. So if you guys look in your notes, what happened in prophase? What was the first thing we said that happened in prophase?
DNA condenses into chromosomes, and each chromosome consists of 2 identical strands called sister chromatids.
Okay, good. So in prophase, the DNA condensed. So what do you think is going to happen in telophase? What would be maybe a word that's opposite to condensing?
It would become larger.
Yeah, it relaxes and lengthens. Good. So the chromosomes relax back into chromatin. What else happened in prophase?
Sister chromatids are held together by a centromere.
Okay, good. So your daughter chromosomes have been separated. We don't need the centromere anymore. The centrioles and the spindle fibers formed in prophase, so they're going to disappear in telophase. The nuclear membrane and the nucleolus disappeared in telophase, so they're going to reappear. Sorry, they disappeared in prophase. They're going to reappear in telophase. Telophase and prophase are opposites. So you have those individual strands of chromosomes at each pole. The spindle fibers and the centrioles, they're not needed anymore, so they disappear. And the nuclear membrane and the nucleolus reappear around each set of DNA. And the DNA relaxes and uncoils back into long, thin strands of DNA called chromatin. So you guys can see that prophase and telophase are opposites. Now, you guys could probably use logic to figure out why these different things have happened. The spindle fibers and nucleolus, they disappear because they're not needed anymore. Why does the nuclear membrane and the nucleolus reappear?
To protect the DNA.
Perfect, to protect the DNA. And why do you think the DNA is relaxing back into long, thin strands of chromatin? Remember, after the cell splits, it's going to go right back into interphase. So why would the DNA relax back into chromatin?
Because it doesn't need to be stretched with the spindle fibers to connect to the other sister, because now they're both separate daughter cells.
Yeah, they don't need to be condensed and held together with the identical copy, but they relax so that the DNA can get replicated in interphase again. Remember, it's long, thin strands so that every portion of your DNA code gets copied and nothing gets missed. So it relaxes again because once the cell splits, it's going to go back into interphase and it's going to replicate the DNA again. So it's important it's in a long, thin strand when it gets replicated. After telophase, this is the end of mitosis. Mitosis is prophase, metaphase, anaphase, telophase, just the nucleus getting replicated. After telophase, then comes cytokinesis, which means cell movement or cell division. In cytokinesis, the cell membrane pinches at the middle. It actually is called cleavage when it's pinched like this picture down here. And then it separates. So it pinches and it separates, dividing into 2 identical copies, and we call those copies of the cell daughter cells. So you started it with 1 parent cell. When it splits into the 2 exact copies, this is a process of asexual reproduction, remember? It's an identical copy of the parent. We call those daughter cells. After the daughter cells split, they go back into interphase. So they're going to grow right away. They're going to replicate their organelles. They're going to copy the DNA, and they're going to wait for the body to say, yes, it's time for you to divide again. Okay. So cytokinesis is the last stage. At telophase, the nucleus is ready to split and the cell's ready to split. It doesn't actually divide until cytokinesis. So here's your phases. This part called M, this M phase is where mitosis and nuclear division happens. Right here is where the cell splits, then it goes right back into interphase. where the cell grows, organelles are replicated, the DNA is copied, and then it goes back into mitosis again, and then it splits. So interphase is about 90% of the time, mitosis and cytokinesis being the last 10%. Here's a very quick computer-generated picture of just the DNA replicating, condensing, organizing, and going to opposite sides of the poles. You can even see it, the metaphase, the anaphase, and then the telophase there at the end. In plants, it's exactly the same. So here's prophase, the DNA is condensed, kind of jumbled up there in the middle. Then it organizes into the middle of the nucleus called metaphase. You can see it's being pulled away in anaphase. And then telophase, it's on opposite sides of my plant cell. But one thing that happens in a plant cell that doesn't happen in the animal cell is when the cell membrane splits and divides, then the cell wall also has to form around each of those daughter cells. So there's one additional thing that happens there. And again, here's just another picture of what's happening here. At the end of interphase, you have a daughter cell. It will go into interphase after it's grown and the organelles have replicated. You have prophase, DNA is copied, sister chromatids are held together, centrioles and spindle fibers have formed, the nucleolus and nuclear membrane disappear. Chromosome lines up in the middle and metaphase. The spindle fibers are attached to each centromere. Anaphase are being pulled away from each other. So one copy goes to one pole, one goes to the other. Telophase, each copy of DNA is at each pole. Centriole and spindle fibers disappear. Nuclear membrane and nucleolus reappear. And then in cytokinesis, it pinches and splits, 2 daughter cells being formed. And then it goes right back again to interphase. Okay.